NICE criteria, so the National Advisory Body in the UK. It takes you through this flag system, so anyone with a red flag moves onto the action tool, and amber flags should prompt further investigation and assessment. So the action tool is very simple. It includes the sepsis 6, the 6 simple actions that need to be achieved within the first hour. So ensuring a senior clinician attends, both recognising the importance and how rapidly women with sepsis can deteriorate, but also antibiotic stewardship, recognising that some senior clinicians may actually be able to step down the concern about sepsis. I won't go through each of the six requirements. They're all very basic: resuscitation, checking bloods, looking for signs of organ dysfunction, and monitoring progress after resuscitation with IV fluids and giving antibiotics. There are some special considerations that we need to bear in mind when managing maternal sepsis. So firstly, the infective organism, the commonest organisms which repeatedly found to cause the most severe disease are E. coli and group A streptococcus, and all empiric antibiotics should cover these according to local sensitivities. And secondly, evacuating the cause of infection, which is often within the uterus, and so recommending early delivery. Maternal sepsis is inherently more complex to manage because we have 2 patients to consider. We must optimise the mother's condition to keep the baby safe, even if this means premature delivery. But sometimes we may also consider whether the baby may benefit from early delivery, thus reducing their exposure to in utero infection. And multidisciplinary care is really essential in making these sometimes quite complex decisions, and should always include obstetricians, neonatologists, intensivists, and microbiologists. The confidential inquiries in the UK sadly suggest that a high proportion of maternal deaths due to sepsis could have been avoided with better care. And there are themes that frequently pop up. And in the latest report, one of the commonest findings was that there was a failure to expedite the delivery of the fetus. And this particularly occurs around thresholds of viability, when decisions are really difficult, both for clinicians and mothers, if we're thinking about terminating the pregnancy for maternal health reasons. Frequently, there's failure to involve senior clinicians early on in decision making, but also failure of senior clinicians to seek help from friends, and looking at more complex diseases and thinking about the infectious diseases specialists getting other specialties to help with the management. So in summary, maternal sepsis is common and a potentially really dangerous condition. There's a big lack of validated data on sepsis in the obstetric population, and we should be really cautious with simply adapting the well-validated tools that are used in the non-obstetric population to reflect what is actually quite poorly understood pregnancy physiology. Meows and screening tools can be really useful in improving the management of sepsis, but they're adjuncts to clinical care and not definitive tests. Maternal sepsis management can be really complex and demands multi-professional care. Thank you very much.
So thank you so much for your brilliant and clear presentation. We have now more than 1,000 participants, so this is an important detail for the success of this meeting. And I would like to ask everybody to send the questions to write the questions, the special session question and answers, and we need your question to have a good discussion. And now, please, Professor Zimmermann, to continue the presentation of the next speakers.
Yeah, it's nice that we reached 1,000 participants. It's slightly higher than in the previous webinar, so that's nice. And let's quickly move on to the next speaker, who is Professor Saverkool. He's Professor of Medical Microbiology, both in Maastricht and in VU in Amsterdam. And he's also the head of the Department of Medical Microbiology in Maastricht. And he will talk about new microbiological techniques to predict and diagnose neonatal sepsis, which is in his specialty, of course. Paul.
Yes, thank you, Luc. I'll try to share my screen. Is it working?
Not yet. You need to click on the green button at the bottom of your screen.
Does Sean has to unshare it or not? Is that done?
Professor Sabekul, do you see
Yes.
the green button or not?
No, I'm not seeing the green button. That's the problem.
See. So do you see only the small picture of Zoom?
Yes.
Okay, so if you click on, there should be an arrow at the bottom left side or right side. If you click on that arrow, it will go to full screen. Do you see that?
No, it's gone.
So if you click on the icon, there should be or the Zoom icon or the image of Zoom that you see now.
For the participants, we did try this before and it worked. So
Okay.
Yeah. Something is coming. Yeah.
Yeah. Sorry for the delay.
Now you have to do slideshow. Yes. Thank you very much.
Yes. Thank you. Well, thank you very much for inviting me to give this this lecture. Sorry for the delay. I will developments, and I will focus on the new developments on that, because I think in the present era of microbiology, we are really in the need of new techniques to improve the treatment of sepsis in neonates. So let's start with a general application. I think you're all familiar with that. When you have a neonate and you have the current gold standard is blood culture. So we normally use 0.5 to 2 milliliters blood from the neonates, which is cultured. And then the microbial is followed by phenotypic identification and then by antibiotic resistance or sensitivity detection. But this whole process still takes from 24 to 96 hours. And that's needed because the levels of pathogens in the blood can be as low as 1 to 10 cloning forming units per ml. Especially when there's fastidious growing organisms, it may take even any longer. So what are the consequences of this current diagnostic process? Is that we also heard it in the previous talk that we use empirical treatment of patients suspected from sepsis. We use broad spectrum antibiotics with all the troubles of getting resistance. And we may even use unnecessary antibiotics. And of course, we have a delayed identification of the pathogen in case of fastidious growth. And of course, we have also the problem that sometimes patients already receive antibiotics, and then this will end up in false negative results in the culture. We all know for many years now that time is crucial in sepsis, whether that's for adults, like in this very old publication, or whether that's in neonates. Time is crucial. If you start too late, then the mortality is increasing dramatically. Still, we are using the same principles as 20 years ago. So what can we do? Well, let's focus on the neonatal. Actually, it's too low for an adequate culture. The immune system of the neonate is, of course, not yet developed, and this has direct influence on the rapidity of the growth of the pathogens. And what we also know that the bacterial load, so the amount of bacteria present per ml of blood, is higher than in adults, and probably because the immune system is not involved in that. So that means that there's a rapid increase in a short time frame, which urge for very short, very rapid intervention. So what we are looking for many years now are rapid diagnostics. And then is the question, what's most needed? Do we need the name of the bug, or do we need antibiotic sensitivity? Well, that's a question which I hope to give you a little bit of insight in today. And do we need new approaches? Do we monitor treatment, or do we even can predict which neonates can develop sepsis? So there are a lot of techniques. I won't go into detail with you of all of them because that will be too much time and also too technical, but I will focus on two specific items, and that's the PCR, the multiplex PCR and the broad range PCR, and on the metabolomics. All the other techniques I will leave out for today, looking at the time schedule. So when we lead at speed, then we have the molecular techniques available nowadays. The advantage of them that they are rapid, that they are very sensitive, and even if there's prior use of antibiotics, that's normally not a big problem. Of course, there are also disadvantages. You only detect DNA. You don't have any antibiotic sensitivity information, and you can only analyze a small volume of blood. But that's normal in neonates, so that's not a big problem in neonates. In earlier days, they were also expensive, but that's no longer the case. They are getting very cheap now, and you don't need any specific lab requirements anymore because the environments are now very used to doing molecular diagnostics in most laboratories, at least. So the question is, we now do culture, but can we skip the culture step? Because if we can skip the culture step, then this will dramatically increase the throughput of the PCR. So what we can do, the volume of the neonatal blood is actually optimal for direct DNA isolation. So we can follow two approaches. Design, on one hand, highly sensitive PCR with a lot of different targets, a lot of pathogens which can be detected, or you can look at broad range PCR. And then we are talking about a time frame of approximately 3 hours, which is of course very very good to have, because then your treatment is at the right time, and you don't need to have broad antibiotics because you can already focus on the real bug. When we use normally in the real-time PCR, which is the normal application in most molecular laboratories now, you use 1 ml of blood for DNA isolation, and you have a numerous number of different PCRs which can detect different bugs. Of course, they include the usable suspects for neonates like Staphylococcus species, Enterococcus, E. coli, Klebsiella, etc. The advantage is rapid result. The disadvantages, as mentioned, limited number of pathogens anyhow, because you can't detect them all like in culture, and you have no sensitivity. So the question is, can we do it better? And then the question is, when we do these kind of PCRs, we also can look at the amount of DNA in blood which is present, which is a direct consequence for the severity of disease. So if you have a very high number of DNA in the blood of the neonates, this also means that there's a lot of bacteria present, either dead or alive, but they shouldn't be present there. So what we did some years ago already, we looked into the bacterial loads in neonates, and what we actually found is that they are quite high compared to adults, for example. And that also means that perhaps we can use that to follow up treatment to see whether treatment is successful or not. So, and if we do that, then we can compare this treatment with other parameters which we already have from the patients. And then you end up with bacterial loads to monitor treatment. And I have two examples here. On the left side, you can see a relative change of Staphylococcus aureus DNA in the blood. And what's nice to see there is that you see an increase of the DNA load, which is the light blue lane. And you see that it's almost the same picture as looking at the CRP, or looking at the leukocytes in the blood. So what you see is that the dynamics of the pathogen DNA in the blood of neonates is as rapid as the CRP development in the blood. This also means that you're now looking not at an indirect measurement, but at a direct measurement on the blood of the neonate and also on the direct pathogen. So you have two things. You have first the direct pathogen, so you really detect that in this case it's an S. aureus who is causing the problem, and you also can see that when you start treatment that the bacterial load is decreasing, and you see an example on the right side where you can see when the treatment is successful that the DNA load in hours time decreases. If this is not the case, then it will increase. So if you monitor that, then we might end up into a direct monitoring of the sepsis in the neonate. Of course, this is work in progress because we have to check a lot of parameters to see whether it's really doing what it should be doing. And the other disadvantage is that we still have to take blood from the neonate, which is of course not what we would like to, because then you want to monitor, then you have to take blood again and blood again and blood again. So that's not really a thing which we think in the future can work out. But on the other hand, can we predict sepsis? Because then we are in front of the problem. So then we can see if there's sepsis suspected or not. And therefore, the focus now in microbiology is on the gut flora, because as one of my former PhD students, Tim DeMay, says, what happens in the gut does not stay in the gut. So what we did is we did a study on 753 infants, which were included. 187 had a late onset sepsis, and we included also more than 400 controls. Looking at the pathogens cultures from these sepsis, you can see on the left there were Staphylococcus, the co-glass negative Staphylococcus were the majority, and then followed by the epidermidis. So, and the gram-positive were there, Enterococcus, Staphylococcus, and the gram-negative with E. coli. When we look at the complete data, we could have, in the end, 40 samples of this late onset sepsis and 40 match control with complete data sets. And that's, of course, important to get really inside information in what's happening there. And when you look at the microbiota composition at its pre-moment, then you can see here until 5 days before the onset of the sepsis, that we can find already the causative pathogen in the microbiota of the gut already present. You can see here the red bars is the gram-positive causative pathogen already at 5 days before onset of the real sepsis. What we can see in the match controls that, of course, they are later present there in a lower amount. So that's also important. On the other hand, we can see that in the match controls, there are no differences on that. And what you can see is that both S. aureus and cows were present in the fecal samples up to 5 days prior to clinical onset. When we look at the gram-negatives, on the other hand, you can see the same picture, but it's even more bars. 3 days prior to clinical onset, we already detected E. coli and Klebsiella pneumoniae are increasingly abundant until clinical onset, whereas absent in all the time points in the match controls. So this does mean that when we see these bugs appearing in the gut flora, we might already start to think to keep that in an eye on it and follow that in time. And of course, that's very more easy to obtain the clinical material than having an invasive blood culture. So in the to summarize that specific approach is that we can see that in the total abundance of causative pathogens, gram-negative late onset sepsis, that already 3 days before the late onset sepsis, we already detect the bug appearing in the microflora. So that's one approach, I think, which could lead to future applications in diagnostics. But we are doing more because we are also thinking about looking to other new molecular methods. So we did also a study together with the Amsterdam, and Tim de Meij was leading that study in 9 NICUs in the Netherlands, and still ongoing. We collected 30-week features daily up to day 28, and we had 2,500 inclusions. So this means that we had 25,000 fecal samples. And we looked at the occurrence of sepsis, and we had the same match controls. And we did, next to microbiota profiling with the IcePro technology, which I showed you also the previous results of, also the volatile organic compounds. So the smelling of the fecal samples. So we did an analysis, and the study population in the end was late onset sepsis in 127 neonates and a match control in which all kinds of different parameters were involved. And the same principle was followed, that we took samples 3 days before onset of sepsis, and we could see that we already could measure something. And the important here is also that when you look at all these specific organic compounds, and I won't go into detail for you with that, it's just to illustrate that when we could make a heat map of 50 most discriminatory metabolites of the subgroups, which significantly differed from match control. So we can really see at the pattern already that something is changing 3 days before sepsis occurs. So perhaps by combining these volatile organic compounds, so the smelling of the metabolic smell print of these children, we can combine that with the microbiota profiling, and we can follow up that we might predict which children will have the risk, at least, to get a late onset sepsis. And I think that is the new way, looking forward to new diagnostics and getting better patient care, and also direct having the right antibiotics to treat with. So this will, if we can follow this up, then this will have impact on the molecular diagnostics in neonatal sepsis. Because when we look at these new data, I think they are quite promising, and we don't have to wait for the culture on that, so we can directly measure that from the microflora of the patients. We also get new insights into disease development, which species are already present, how do they get there, what do we learn from that. And we might even use bacterial loads for monitoring treatment, as mentioned before, even in the absence of antibiotic sensitivity. So I think also that the potential of fecal volatile organic compounds, microbiota, and metabolomics, we might go into a new era of preclinical prediction of late onset sepsis, which we also can use for NEC, of course, but that's an item which I won't discuss today. And I think very important, we can make use of non-invasive biomarkers, which is, of course, also for the patients itself the best way to deliver samples. And now, when we can develop that, and it might even end up in possibilities toward point-of-care testing, because the techniques are quite easy to do, especially this FOX analysis, which is very easy to do in a laboratory. So this is where I want to leave you. I hope I have given you an impression of the new developments also on the microbiology field in detection and diagnosis of sepsis. And I want to thank Tim de Meij and Dries Benning, 2 former PhD students of mine who are taking it up in their own groups now, which I'm very proud of. And of course, all the other collaborators, including the NICUs in the Netherlands. And I want to thank you especially for your attention. And I hope you have if you have any questions, I will be glad to answer them. And it's a little bit weird to talk to my computer all the time, but that's the only thing which we can do in these days. Thank you.
Thank you so much, Professor Saverkool-Pohl. Thank you. It was a great lecture and a good look into the future. So you were proud of your previous PhDs, I'm also proud of one of mine, who is the next speaker, and that's Alessandro Borghese. He is a neonatologist in Pavia in Italy, and also has an appointment at the School of Life Sciences in Lausanne in Switzerland. He is a neonatologist, but he's also mainly into infectious diseases. So he will give his talk on neonatal infections, genetic and immunological insights. Alessandro, the floor is yours.
Okay, can you see my presentation?
Yes.
Yes.
Okay, so first of all, thank you very much, Luke, and also Vassilios Fanos and Elias Saliba for the very, very kind invitation. It's really a privilege for me to be here. And I will talk about the other side of the infection. We just heard about the microbiological side. Now we will talk about the host side of infection. So specifically for this infection, I will focus on severe life-threatening infections occurring after after first exposure during postnatal life, during the first weeks of life. I will not focus on congenital infection. And to understand where we are in the field of neonatal infections, we go quickly and step back to the history of infections and go to the 18th century, when scientists were even asking whether infections came from inside or outside the body. And the answer to this question came the century after, with epidemiological studies and microbiological studies demonstrating incontrovertibly that microorganisms cause infection. So infections come from the outside. But soon after, in the beginning of the 20th century, scientists started to understand that microorganisms did not explain it all, and that they were necessary to cause infection, but they were not sufficient, and that there were factors, host factors, that were implicated and then influenced the final outcome of an infection. In the 1950s, there were the first discoveries of the first primary immunodeficiencies that were sound conditions with recurrent life-threatening infections by a wide range of microorganisms, eventually leading to death. These were conditions that took the attention of physicians and scientists, but were still considered a niche compared to the wide field of environmental infections that came from outside the body, essentially almost all infections. But then the field focused on a narrower spectrum of susceptibilities, and eventually in the past 2 decades there was what we can call a true revolution, because attention of scientists focusing on susceptibility to infection went into common infections. And what we do know now is that common infections like herpes simplex encephalitis, pyogenic bacterial infections, influenza pneumonitis, and even in 3–4% of cases, COVID-19 pneumonitis are single gene disorders. That is, single gene disorders that cause impairment in the immune response and that eventually lead to susceptibility to life-threatening infectious disease. So what has happened in the past 2 decades has been the filling of a gap that existed between the small niche of primary immunodeficiencies and the wide field of environmental infections, and this was filled with the recognition of single gene defects leading to even single pathogen, single episode infections. That is what we classically consider environmental infection. So all this is quite acquired in the general field of human infections, but is a bit less known in the field of neonatal infection. So how can we translate this for neonates? So first consideration we need to do is that neonates are not all equal. We do know that that when neonates get infected, some of them were previously sick and some of them were previously healthy. And likely the pathogenesis of infection in these two groups of infants is quite different. So let's focus first on previously sick. Let's take preterm infants as a paradigm of this group. And if we consider preterm infants, we do know that they are exposed to an array of factors: antibiotics, surgery, central lines, admission to an ICU itself. And in this context, the role of genetics is likely a minor role. This can be guessed just by observation, but this is what we know from studies that clearly demonstrate that the role of infection is small in the pathogenesis of infection. The role of genetics in the pathogenesis of infection in preterm infants is very small. A genome-wide association study was conducted a few years ago. This was a great idea. The point is that it was likely not sufficiently powered to detect very small effects of genetic variants on the outcome sepsis yes or sepsis no. So the conclusion we can draw from this first data is that at the population level, there is likely a minor role of genetic factors, or alternatively, genetic factors have small effect sizes in influencing the occurrence of infection when we talk about preterm infants and more in general about previously sick infants. If we then focus on full-term infants, they can get infected as well. And we all know that they can have serious infections, even lethal infections. And in these infants, infection is still an enigma, is still a mystery. So we don't know why they get infected. And a response comes from population-based studies of the neonatal immune system. These studies have their roots in the studies in the 1940s and 1950s that discovered what is now well known as the fetal tolerance, and then gave rise to the concept of neonatal tolerance. But afterwards, starting from the 90s, several studies that are hardly summarized in this slide led to the concept that neonatal responses can be protective, but are quite different from adult responses or immune responses in childhood later on in life. So from these studies, the main acquisition, the main concept we have is that they do explain at the population level why neonates and young infants are more susceptible to some infection, have a higher incidence of life-threatening respiratory infections or group B strep-like neonatal pathogens. But they don't explain why they're fully resistant to many pathogens, some of them opportunistic, but some of them even virulent. And more than everything else, they don't give us back a missing piece, that is the explanation of the wide inter-individual variability in clinical manifestations. So studies on the neonatal immune system does not explain why, once exposed to the same microorganism, one infant will die and another will stay totally asymptomatic. So to try and give you a hint of what we know now, I brought the example of bronchiolitis, and we saw from one slide of Xian Edwards that lower respiratory infections are one of the main causes of mortality globally. And in terms of public health, bronchiolitis is a very important issue. But when we go and see in terms of proportions, we see that a small proportion of all infected infants, of all exposed infants, actually need advanced respiratory support, CPAP, mechanical ventilation. A very, very tiny proportion has a little infection. So severe infection is a rare outcome of bronchiolitis. So what can we say about bronchiolitis? We cannot explain all cases, of course, but some cases have an explanation. And what we know is that cells can recognize the viral nucleic acids through a receptor, that is MDA5, that once it's activated can induce the production of antiviral interference and induce a transcription program into the cell that leads to cell intrinsic protection, cell intrinsic immunity. The cell is protected itself from viral replication. But if this receptor is mutated, antiviral interference are not produced and cell intrinsic immunity is not provided. The virus can replicate itself and infection progresses to clinically relevant severe infection. This has been demonstrated in at least 2 papers, showing in one case, one infant with recurrent infection by human rhinovirus, and another paper showing single episode infections caused by respiratory syncytial virus or human rhinovirus in young infants. So one conclusion we can draw from this, the knowledge of this mechanism, is that a first infectious episode in a neonate or a young infant can be the first manifestation of a primary immunodeficiency. The infant will go on at follow-up having multiple recurrent infection, possibly there is a family history. The infant will go on immunological testing, genetic testing, and a diagnosis of primary immunodeficiency will be done. But this is one of the most rare cases. In most cases, infections in newborns and young infants are sporadic. There is no family history, and they are single episodes. At follow-up, infants are healthy. So the question that is still unanswered for most cases, but as said, we have a hint from the example of bronchiolitis, are these infections, first infections in neonates, more genetic than we think? Are there mechanisms that we are still not recognizing? And if this is true, are we missing the opportunity to provide to infants a molecular diagnosis? So the way we think the field is going is this one. We think that in neonatal infections, we still need to discover single gene disorders that will explain infection in full-term infants with no comorbidities. So how to address, in general, the problem of individual or population susceptibility to infection in neonates. So current techniques, current technologies allow to address these problems. And one way to address these problems are genome-wide association studies. We have seen that there is just one single genome-wide association study performed in preterm infants, but this is not a reason not to go on with this kind of studies. The only point is that we need to identify well measurable outcomes. We need to sufficiently power the studies, and we need to well define the study phenotypes. For example, we need to go deeper in phenotype definition. We can use other omic techniques like metabolomics, for example, to identify phenotypic patterns of metabolites, for example, and we can address the question of common variants influencing the pattern of metabolites. So genome-wide association studies will be likely very useful. We try to predict the future, and our prediction is that GWAS, genome-wide association studies, will likely be very useful in identify the role of the human genome in modifying and influencing some patterns of infection, some variables, some phenotypes. On the other hand, we also need sequencing studies, exome sequencing or genome sequencing-based studies that target not populations of infants, but single infants or small cohorts of infants with well-defined phenotypes. These studies are able to identify rare variants that explain extreme phenotypes, identify pathways that are affected in infants who suffer from life-threatening infection, and ultimately are able to lead to the explanation of why individual infants will suffer from a life-threatening infectious disease. So with this, I conclude my presentation. And just to summarize, we know now that common infections are single gene disorders much more frequently than we thought 20 years ago. And also neonatal infections can be genetics, and they can be the first manifestation of a classical rare primary immunodeficiency, but likely many neonatal infections will prove to be single gene disorders. Not all, likely, but many will prove to be single gene disorders. In preterm infants, we need to use other types of approaches, always thinking about the marginal role of the genetics in the context of sick infants, and so well powering the studies. And final sentence is that just we need to combine the available technologies, genomics, metabolomics, and also microbiomics to answer to the open questions. And I would like to thank all the people working with me inside my hospital and outside the hospital, and all the collaborators, and thank you all for the attention.
Thank you very much, Alessandro. Very nice overview. Also look into the future. So we are going to the discussion now. So we will look at the questions that are asked in the question and answers. You still have the possibility to ask questions, but we are slightly late, and we of course want also to give the opportunity to the two short presentations at the end. So we have about 10 minutes for the discussion. So Vasilios, you also looked at the questions, so maybe you can start with one of the questions you want to ask to one of the speakers, and then I'll continue.
First of all, I want to thank all the three speakers for this exciting session with the cutting-edge research, and we are looking to all data with new eyes, the eyes of omics technologies from the Greek word oma, which means an holistic approach, metabolomics, microbiomics, and genomics. And I will add to this also artificial intelligence. But let's start with some questions for the first speaker. We have a question from Emrieta Bayrami. Do we need hemogram after 12 hours in mothers with leukocytosis?
I don't think there's a yes and a no answer to that question. I think the risk of sepsis tool that I demonstrated in my talk from the UK Sepsis Trust would place leukocytosis as one of the risk factors, and I would simply look through that tool and decide how worried I was about the patient. Are there any other concerns that actually should trigger further investigation sooner? Are we actually worried about organ dysfunction with sepsis? I think if you find it as an isolating finding, then repeating it later on is definitely a good idea. But I think you can't just take the leukocytosis alone as an indicator. You need to look at the whole picture.
Would you use other screening things like CFP frequently, or? Yes,
yeah, no, I think in the Sepsis 6 package, if you were concerned about sepsis, you do a full blood profile. So doing a lactate and look at full blood count, as well as your renal function, liver function, coagulation, and see if there's any other signs of organ dysfunction that would actually make you more worried about sepsis.
We have another perinatal question by Ibrahim Boucharaf. Is positive placenta culture or histopathology changing the decision of duration of antibiotic treatment in the newborn?
I don't tend to get involved with the treatment of the newborn, but in terms of obstetric management, then these swabs usually come back much later than are useful for us. Obviously, if they come up with something that hasn't been covered by our empiric antibiotics, that's really important and might trigger further treatment. But I'll hand over to my neonatal colleagues on how long they would treat the baby.
Who would dare to answer this?
Yeah. Do you want to add a comment, the other speakers?
I would say that what we should do is to compare what is going on in the infant and how are the cultures in infants, and compare blood cultures from the infant and that from the placenta, and see if they match, and see how is clinically the baby. If she is improving, then maybe we can go on with the same treatment, and if not, we can change according to the culture. I don't know if Paul agrees.
Yes, I agree completely. That's I think there are not so much choices on that, actually. So that's, I think the clinical perspective is very important now, looking at the infant and see whether he or she is improving. So therefore, I also think we should use better diagnostics to see really what's happening, because we don't know what's happening. What is this treatment doing? Are we really treating the right bug? Is there something else going on? We only can see at the infant whether there's improvement, and then no doctor wants to stop treatment because you see that you're on the right way. So please continue. That's more or less the adagio on that. So I think that's the current practice.
I think from a pragmatic perspective, it's really useful that the mother and the baby are a continuum, and that we've got to make sure that our communication between the obstetricians and the neonatology team is good, because if we've picked something up on a maternal sample that we've taken in labour or immediately after the birth, and we're getting that result back 3 days later, that could be very useful information for you for managing the neonate. And conversely, sometimes if you're picking up infections on the baby, then we need to know that as obstetricians if we're still managing a mother that is unwell, for example. So I think that although sometimes the labour wards and the neonatal units are separated in geography, that actually it is, once that umbilical cord is cut, shouldn't stop our relationships with each other, because information that we have as obstetricians may well be very useful for the neonatologists.
What I want to say is that Jo Kovs is also an obstetrician in Bristol. She was the only one that we didn't introduce so far yet, and she's part of the panel. So thank you for your comments. So the next two questions are related, I think. It's, is it necessary to take an anaerobic culture on neonates, or did you get blood cultures from neonates after delivery or later? That depends, I guess, but...
Yeah, that depends, of course. It depends also on the clinician and the suspicion of what we get in, because it's not only up to us what we get. And sometimes we get only aerobic, and sometimes we get both. And that's also why, as you saw also in these studies, that a lot of collections are not the data complete, because then sometimes you get anaerobic and aerobic, sometimes not, and then you have to make decisions, and this will reduce your actual amount of completed data. And concerning the blood samples, that's also difficult, of course, because we don't want to draw any blood from neonates when it's not necessary. So in practice, our T0 point is always when there's a clinical suspicion on sepsis, or when there is fever or whatever, with something going on. When the neonatologist thinks we have to take a blood culture. At that moment, we start also our study, and then it's a normal follow-up, because we want to gather more info based on the routine diagnostics. So, and we want to learn from that, but it's not allowed, of course, ethically, to draw a lot of blood from these neonates. So that makes it very complicated, but I think we can manage. So the time 0 point differs from one infant to the other. That's
what I want to say is that we have these two short presentations at the end. They both go into that, because they look both at predicting the risk of an infection and when to handle, one with a heart rhythm and the other one with a sepsis calculator for early and for late sepsis. So we'll also have two presentations on when to go for it, let's say. Alessandro, you wanted to say something as well?
Yes, okay. So I think that we just sorry, was just reading the other question, so I was not really listening to what you were saying.
No,
sorry,
okay.
Apologies. So sorry. So let's
Okay.
go on, please.
Yeah. Julius, you want to take another one?
Another one question, yes, for Alessandro. Are there is genetics associated with ethnicity? One question. And are some genes associated with early neonatal sepsis? These are two questions of the audience.
Okay, so yes, so of course, genetics can be associated with ethnicity. It is hard to see this in single gene disorders. That is, single gene disorders are usually conditions that are when they cause life-threatening infection, they they cause lethal diseases, and they are rarely selected for in populations. So I would say that we don't have to think about ethnicity influence on rare conditions causing life-threatening infection, while when we do study population, we do population genetics, and we do genome-wide association studies of some outcome, infectious outcome, then we have to take into account ethnicity, because some ethnic groups may be different, and we could see differences just related to the ethnic groups. Hope to have answered this a big question about yes, ethnicity, so hope to have answered to this. And the other question was about genes for implicated in early onset sepsis. So of course there can be, I do know about two reports, one about Kostman neutropenia and another on chronic granulomatous disease causing early onset sepsis, lethal early onset group B streptococcal sepsis. So these are reported, are available in PubMed, one 1988 and another one in some years later. And the point is that there are no systematic studies of full-term newborn, of genetics of infection in full-term newborn. So we don't have available cohorts that have been studied. So this is something that we will need to do in the future.
Thank you.
I ask a question to Alessandro. Do you have any sex effect on susceptibility in these newborns?
Okay, so if we go and see genes and we there can be X-linked disorders that can influence. If we always need to make a distinction between rare conditions, rare genetic defects causing life-threatening infection, and common defects causing sepsis in sick infants. So if we consider rare conditions, the only influence I would see is related to X-linked disorders. While if we go and see other mechanisms that are influenced but are not so deterministic, so influenced by the human genome, then we can likely see an effect of sex. For example, it is very shortly, we do know that there is a bias that men are much more affected than women in COVID-19. They're not neonates, but there is an influence of sex. And we do know that this influence is related to the production of autoantibodies that block interferon responses. So men are more prone to produce some blocking, some neutralizing autoantibodies that impair their immune responses than do women. And similar mechanisms may be working in neonates, but this is just speculation. So we do not know for neonates.
I'm afraid we are running out of time a bit. I don't know, I asked MCA how strict the end time is, but we really want to stop about in time, and we are a little bit late. So I asked the speakers, the three previous speakers, to answer some of the questions still written in the questionnaire, and then we can move on to the 2 short speakers. Otherwise, we're gonna really run over time. So, and if we have some time at the end, then we might still discuss a bit. Vasilis, you want to introduce the next speaker?
Yes, it's my pleasure now to introduce Daniel Keane from UK, and the talk is about early onset neonatal sepsis, evaluation of the Kaiser Permanente tool. Please.
Hello, can you see my screen?
Yes.
Yep. So hi, good evening. My name's Daniel. I'm a paediatric registrar based at Medway Maritime Hospital in Kent in the UK. It's a tertiary neonatal centre. Just want to start by saying thank you for having me this evening and to present my abstract. It was the project that we undertook to look at the Kaiser Permanente sepsis tool and its applicability to our local population. So something that we've noticed over the past few years is that we're getting a higher volume of well-appearing babies being treated with IV antibiotics for suspected infection. Probably the most common reason for this is suspected infection in their mothers or mothers who are presenting with high fevers in the peripartum period. You would have heard earlier on in one of our previous talks that there's been quite a lot of teaching in the United Kingdom the last kind of decade about sepsis, and it's coming to the forefront. So our thresholds for treating suspected infections has decreased. And unfortunately, with national guidance, if we're treating our mothers with broad spectrum antibiotics, we also treat the babies. However, having said that, the actual incidence of early onset neonatal sepsis remains low. So problems with kind of broad use of antibiotics in our neonates. So we are getting some reports, particularly in the United States, of resistant strains of organisms, particularly those that affect neonates. And a hot topic of debate at the moment is the alteration of the gut microbiome from using broad spectrum antibiotics in our newborns and the long-term effects that might have. But possibly more personally this year with the COVID-19 pandemic is that we've had to be quite restrictive on our visitation policies on our wards. And keeping mothers with their babies in hospital leads to separation from the family unit. And we've seen this firsthand that this can cause quite a lot of stress. So when we look at the Kaiser Permanente tool, it was a multivariate algorithm that was designed based on a large observational study in the US. And when they implemented this tool, they demonstrated a dramatic reduction in the number of babies receiving IV antibiotics, but with no associated rises in adverse effects or missed cases of actual sepsis. If anyone is unfamiliar, this is what the tool looks like, and it's freely available on the internet. It requires you to put in some local demographic, but also maternal risk factors, such as the duration of the rupture of membranes and the highest temperature during labour. Once you compare that with your clinical examination of the infant, it will then give you one of 3 risks of infection. So for example, if you had a well-appearing baby, your risk of infection would be 1 per 1,000, and then next to it, it would give you a recommended management plan. So based on those findings, it's attracted quite a lot of attention in the United Kingdom. So other centres across the country have done similar sort of studies and managed to replicate effectively these results. Some centres have actually now implemented the Kaiser sepsis tool as part of their clinical practice and are seeing some very positive findings. So we were quite keen in Medway to see if it would be applicable to our local population. So we designed a prospective observational study where we collected data from infants that were screened for infection based on our national guidance. These babies were mostly started on antibiotics after delivery. We then followed up their biochemical responses and their culture responses and compared that with the recommendation plan that they would have got had we been using Kaiser. We recruited 62 patients to the study who on average stayed in the hospital around 3 days on average. And you can see the most common cause for starting antibiotics in these infants was maternal fever in peripartum period. So looking at their biochemical responses, so we stratified this based on CRP. So around half of the infants had a low inflammatory response of less than 5. And a quarter each in the moderate and the higher risk categories. But it would be important to note here that all of the blood cultures and CSF cultures that were taken returned negative. We then clinically examined them. So in the green boxes, that indicates that the baby was well appearing and examined well at the point of screening, and yellow would indicate that they had some abnormalities on their news or early warning score charts. And then this is what the Kaiser sepsis calculator would have recommended at the point of starting antibiotics. And what you can see is there's a dramatic shift away from kind of immediate IV antibiotic treatment with a favour in serial observation and serial assessments. And what we can't show from this study, unfortunately, is how many of these babies who, if we had chosen not to start antibiotics but to observe, would have ended up needing antibiotics through a change in their vital signs, because obviously all of these babies were on antibiotics at the point of entering the study. But nevertheless, we were quite optimistic that we'd be able to replicate a similar reduction in potential antibiotic use in our well postnatal babies. However, we emphasise that this obviously requires quite high clinical vigilance, staff training, and getting familiar with regular routine checks of the baby whilst they're on the postnatal ward. So we are naturally quite excited about the idea of possibly reducing the amount of antibiotics that we could use. And that was my abstract.
So, thank you so much. Please, Luke, continue with the last speaker.
Yeah, thank you. And thank you for the keeping to the time, because you have, so we'll move to the last abstract, which is from Patrick Pladis from France. In very preterm infants, bradycardias associated with late onset sepsis differ from bradycardia associated with immature breeding. So it's about predicting the sepsis, let's say, by the bradycardias. Patrick.
Okay, do you see my slide?
Not yet. We see a black screen.
Okay, sorry.
But it's busy loading, I think, because no, it's gone. Yeah.
Yeah, okay.
If you start the slideshow, then you can. Yeah.
Yep.
Is it okay?
Perfect, thank you.
Okay, sorry. So my name is Patrick Pladis. I'm professor of neonatology in Reims, which is in the western part of France. And thank you to give me the opportunity to present some of the first intermediary results from the European DGNUB project. This project is the consortium of this project included the 6 university hospitals and engineers from 3 universities and 2 SMEs. Very preterm infants are especially vulnerable, with 10 to 25% of them presenting at least one episode of late onset sepsis, which in turn increases the length of stay, morbidity, and mortality. Studies have found that prompt diagnosis and administration of antibiotics can significantly reduce mortality. However, diagnosis is difficult to ascertain at an early stage. Indeed, all the elements of diagnosis, whether clinical, biological, or microbiological, are usually unreliable or discrete, non-specific, and late. That leads to over or late administration of antibiotics, which is suboptimal. The objectives of the DGNUB project was to build a decision support system for real-time, non-invasive, data-driven, standardized, explainable, and user-friendly system. This was done using a multiparametric analysis of variability of high-resolution vital signal, analysis of video and sound, and clinical signs. We included more than 500 newborns with the objective of late onset diagnosis in the cohort. Among them, we had 48 infants, preterm infants less than 30 weeks of gestation, having received more than 5 days of antibiotics beginning at least 72 hours after birth. we had a control group of preterm infants who never received any antibiotics. The first step of the study consisted to work on the definition of late onset sepsis in the late onset sepsis group. And the first constatation was the number of validated late onset sepsis cases varied widely according to definitions. We have 13 patients according to CDC criteria, 28 for NICHD, 41 for NeoKIS, and 46 for Canadian Neonatal Network definition. The low Fleiss kappa coefficient indicate a very poor inter-observer agreements. The best concordance between a panel of 5 French experts and international classification was obtained for NeoKIS, which was consequently used as a definition for the diagnosis of sepsis. The figure at the top presents the time window used for analysis of late onset sepsis. And you have, for late onset sepsis, the sepsis period and the control period, which is more than 5 days after the T0 time. And in control period, there was a randomly selected matched period. And what you can see is that the population the subpopulation were comparable in a case control approach between late onset sepsis and control group with a start of antibiotics around 8 days of age. We analyzed 5 parameters about bradycardia events. Bradycardia was defined as less than 100 beats per minute for more than 5 seconds, and they were characterized. And the main results of this characterization was that the number of bradycardia was higher in case of late onset sepsis than related to immaturity, and then the mean interval between the events was shorter for late onset sepsis than for the control population, and with a difference that were also visible in the slope of decrease in heart rate and in the distribution of the duration of a bradycardia. After this, we did a discriminant analysis using a late onset sepsis group versus control group. And we test this, the formula obtained between before and after sepsis. And what we obtained was a predictive positive value of 71% and negative predictive value of 68%. We also worked on heart rate variability results with recurrent neural network, which is an artificial intelligence-based method which takes into account the time. Diagnosis was relying on heart rate variability, which have the advantage of being non-invasive, readily and continuously available in the context of neonatal intensive care units. And moreover, it's an evaluation of organ dysfunction. We had the first talk about the definition of sepsis. Heart rate variability was used in the 3 different categories of features: time domain, frequency domain, and nonlinear measurement. And we also used a new method, which is visibility graph analysis. And from the first step, using the extracted features, was to select a set of features for the training set, then to build the model using artificial intelligence. Then the algorithm was trained and it was tested on a test set, and the diagnosis value was estimated on an area under the receiver operating curve by 6-hour intervals. And the graph at the bottom presents all the hour curve values evaluated on a sliding window with 6 hours, with a 3-hour overlap between T0 and T0 minus 48 hours. And you can see that we get an area under the curve greater than 0.85, 18 hours before the beginning of antibiotics, with a top value around 0.9. In conclusion, differences exist between bradycardia associated with late onset sepsis and bradycardias associated with immature breathing. Analysis of bradycardia can therefore help for the diagnosis of late onset sepsis. The proposed decision support systems offers the potential for continuous, non-invasive, data-driven time monitoring of the risk of late onset sepsis in a NICU setting. Thank you.
Thank you very much. It was really nice, and it offers a lot of potential for the future. I don't see Vasilios anymore. Vasilios, are you here? I'm afraid that we lost him, so I will close the session then. So thank you to all the speakers. It was an excellent session, and I'm very grateful to you all to give these lectures, and also the two abstract presentations were really complementary, I think, to the other presentations. Thank you so much. Also to our audience, who was over 1,000 people, decreasing slightly towards the end. But I'm sorry that we couldn't all answer all the questions live, but I hope you got most of the answers also in written. I think we can promote the next webinar that you see here, which will be on April the 13th on diabetes and obesity, of course, obstetric related, not in general. So thank you all. It was great, and see you